everyone, and I'm here again with a new video presentation for you all. The topic of my presentation is ginsenoside RB1 attenuates lipopolysaccharide induced chronic neuroinflammation in mice by tuning glial cell polarization. It is from Journal of Traditional Chinese Medical Science and its impact factor is 2.6. My name is Jyotirmay Pradhan under advisor, Professor Zongrong Sir, and I'm working under the lab Neuroscience and Naturopathy lab. Next, I have my contents. First of all, I would like to share something about myself, and next we will discuss about my seminar report, under which it comes introduction, aim of the experiment, keywords, abstract, material and methods, results and discussions, and conclusion. So my name is Zheng Mei, given by my university, CYCU, but usually students and my lab mates call me JP because I have a long Indian name, Jyotirmay Pradhan. And my birth date is July 5, 1995, and my native place is from India. And now I'm living in Badal, Thailand city. My hobbies are singing, sometimes listening music, dancing, and go upon hiking and also camping, and do some artwork sometimes. My zodiac sign is Virgo. So here are 4 dishes you can see. So first 2 are my new interest Taiwan dishes. That is teppanyaki and hot pot. Also, I would like to tell you that I am a pure vegetarian, and these are the Indian dishes I like to eat every time. This is samosa and this is pani puri. This page tells us about my educational qualifications. That is, I have completed my bachelor's and master's from India in the Department of Life Science Geology. My research field here is in the Department of Biomedical Engineering, CYCU, to know upon the effect of 3,5-DMA loaded liposome against non-small cell lung cancer. And finally, I would like to introduce my husband. That is, I am married and he is working in Kwaishan, Taiwan. And he has also completed his PhD in thermal engineering. Introduction. Neuroinflammation is an important factor contributing to cognitive impairment and neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, Huntington's disease, and many more. So as we are talking about the use and the effect of ginsenosides, so the ginsenosides have these many forms: F2, RG3, APPD, RH2, CK, RD, GYP17, and 75. But we will be talking about the RB1 form. So it has various kinds of useful effects. That is, it is anti-diabetic, anti-cancerous, immunomodulatory, cardioprotective effect, neuroprotective effect, sexual endurance. It is anti-allergic too, anti-aging. It also gives anti-inflammatory effects, which we will learn in this study. And lastly, hepatoprotective effect. So the study developed a chronic neuroinflammatory model in mice with an escalating dose of peripheral LPS, that is lipopolysaccharide administration, in an attempt to mimic or copy the chronic inflammation model in the neurological diseases. So here is the aim of the study, that is to evaluate whether ginsenoside RB1 can attenuate lipopolysaccharide induced chronic neuroinflammation in mice and to explore its relationship with glial cell polarization. That means it is telling us about that how this ginsenoside RB1 can regulate or control the LPS induced model of mice and regulate the glial cells. Next, we have the keywords inflammation. What do we mean by inflammation? It's a common pathophysiological process in several neurological diseases. A controlled inflammatory response can be beneficial by promoting tissue repair and removing harmful debris. However, a sustained and uncontrolled inflammation cascade is likely to be damaging too. Consequently, inhibiting or attenuating inflammation is an important strategy for neuroprotection. Next, we have ginsenoside RB1, which is a triterpene saponin that was first isolated from root of ginseng, Panax ginseng. RB1 has various effects that we saw just now with a very good picture. So Panax ginseng is one of the most commonly recognized herbal medicines in China. It has been used in clinical practice for treating numerous diseases, especially neurological disorders, either individually or in combination. Next, we have microglia. These are the resident macrophages in the brain which plays protective function to remove cellular debris and eliminate toxic metabolic materials. However, excessive and prolonged activation leads to neuronal death and pro-inflammatory cytokines. So glial cells or neuroglia are cells that surround the neurons of CNS embedded between them, providing both structural and physiological support for its smooth function. So in this study, we must know what actually are glial cells. These are not the neuron cells. These are the surrounding cells of the neuron, supportive and the protective cells of the neuron, which are along with neurons always. So their regulation is important, which is seen in this study. Next, we have this picture or flow chart showing us about how there is or what are the reasons that creates a systemic inflammation in an organism. Systemic inflammation occurs when the immune system is constantly defending the body, that is protecting the body. The immune system is constantly working. But at some times, the stress, infection, or chronic diseases can put the body in a pro-inflammatory state. So when this happens, at this point, the immune system becomes primed, that is charged, and ready to create an inflammatory response, which is known as systemic inflammation. So this is caused by two types of molecules. There is pro-inflammatory molecules and anti-inflammatory molecules. From its name only, we can see or get to know that the molecules which cause inflammation are under this region, and those molecules that regulate inflammation or reduce inflammation in the body, anti-inflammatory. So these are the molecules. So pro-inflammatory molecules are TNF alpha, IL beta, INOS, IL-8, interleukin 12, and CCL2. Similarly, in anti-inflammatory molecules, we have interleukin 10, tumor growth factor beta, EGF, VEGF, and CLCF1. But here we can see the highlighted region, which shows that these molecules act as both. That is IL-6, arginase 2, and arginase 1. So sometimes these act as pro-inflammatory and at other times they act as anti-inflammatory according to the need of the cells. So this slide gives a brief idea about what are glial cells and how many types of glial cells we have in our body, in our brain. So there are 4 types of glial cells named as microglia, astrocytes, oligodendrocytes, and ependymal cells. Next, microglia have 2 phenotypes. Similarly, astroglia also have 2 phenotypes, M1 and M2, similarly A1 and A2. So the markers that are used to know the level or the situation or the increase or decrease of microglial phenotypes are INOS, arginase 1 for M2, C3 for A1, and CLCF1 for A2. And there is a very good relation we can see upon here that they act antagonistically, that is alternatively. That is M1 and A1 act as killer type cells and M2 and A2 act as repair type cells. So these are the phenotypes and markers of glial cell polarization. Two to three tests were done with the affected or the chronic inflammatory model mice to know the activity of the mice, if it has declining effect or a positive effect. So open field test is done to access exploratory behavior and anxiety in rodents to see if it is anxious or it is free to move anywhere without any hesitation. So it is done in a box of each unit made up of 45 into 45 into 50 per cube. Before experiment, mice is acclimated to OFD unit for 1 minute. Then a 6-minute test conducted every time on 14th day of the experiment to know the movement, distance, and duration, number of crossings, and average velocity. So these are the data that are collected by doing the open field test. Next, we have another test done named as beam walking test. That a mice is left over here and with a given amount of time, it is marked that if it is crossing this level properly or not. To assess motor condition and balance, yes, conducted over 3 consecutive days. 2 days of training is given, and in the last day for test, 12th, 13th, 14th day in this experiment. So these are the data that are collected from this test. There is food slips, how many, and it falls how many times if the mice traverses the entire beam or not normally. So this is the experimental setup of the study. So total mice taken are 56 in number, male mice. It's divided into control, LPS, RB110 group and RB120 group. That means control has 14 number of mice and it is administered with saline solution only. Next, we have LPS group that is administered only with LPS. And the number is 14. Similarly, other two also. By the RB110 group, that is the ginsenoside group that are given or administered 10 milligram per kg with LPS, alternative days. Similarly, in RB120 group, 20 milligram per kg with LPS is administered. So this is the experimental process. So here we can see the mice, that is the chronic inflammatory model of the disease that it has acquired to treat, to know that if by administrating the gene, ginseng RB1, so 7 days of adaptive feeding before the treatment or before the administration of LPS and saline. So here in the morning, it is weighed every day, and LPS is given intraperitoneally. The injection is given. Slowly, we can see the LPS amount is increased 2 milligram per day, 1 to 3, then 5, then 7, then 9, then 11, then 13, up to 14 days. On the last day of administration, the tests are done, OFTs and beam walking tests. And the ginsenoside RB1 or sterile saline at 8 p.m. every day is given to the mice. So before discussing the results and the discussions, we must know about the tests which are being done to get the data properly. That is, the Western blot analysis showed that the nuclear factor kappa NF kappa B signaling pathway was activated in the LPS groups. We will see in a few minutes in the further slides. LPS given to the mice was dissolved in sterile saline, aliquoted and stored in -20 degrees Celsius. RB1 also dissolved in saline, sonicated and stored. After a week adaptation, the mice were divided into 4 groups that we saw now. The mice except in control group were given escalating doses of LPS from 1 to 13 milligram per kg with a 2 mg per kg increase each time that we just discussed now, intraperitoneally at 8 a.m. every 2 days for 13 days. RB1 is given intraperitoneally too at 8 p.m. but at night for 14 consecutive days, while control and LPS group were given saline. The body weight and food intake of mice monitored every day. Anesthesia used pentobarbital sodium 50 milligram per kg intraperitoneally while doing the qPCR experiment. And the blood samples collected from retroocular venous plexus, that is eye region, to collect the data. And hence the results showed that systemic chronic inflammation with obvious behavioral abnormalities was successfully developed in LPS mice, which could be significantly eased by RB1 treatment that we will discuss by discussing these results. So here we can see that the first slide of the result shows us about the effects of RB1 on the body weight and food intake of LPS injected mice. So the first graph shows that initially there was a decrease in the weight of the mice body, but significantly it came to a certain level and there was no much disturbance in the weight of the mice body. Similar case in the case of average food intake. The intake was slow in the beginning, but gradually it was levelized and there was no much hindrance between low and high food intake. Women control and other groups. Next slide shows us that the spontaneous activity monitored for 6 minutes using the open field test after the last RB1 treatment. That means here we can see the pathways of the mice. Control has a very rapid pathway that it can move very freely and very frequently. Here we can see there are lots of lines, but in the LPS group, the LPS administered mice is so dull that it is unable to move or find pathways correctly to have a good movement or a balanced body. So there are no more pathways, very least pathways we can see here very clearly, which shows us that LPS induced mice is the chronic model mice, which has the disease and has the neuroinflammation. But when there is slow administration or the increased dose of RB110 and slowly the RB120, here we can see the pathways is increasing slowly. And in RB120, similarly, the pathways are more, increased number of pathways, so that we can see that the mice is recovering with the disease of neuroinflammation, the inflammatory effects are reducing. So these are the graphs. Here we can see the total distance covered is least in this LPS group. Moving duration is also, it takes more time to move. The number of crossings are least in LPS. Here we can see this blank picture. And the average velocity is also least in LPS. And in all RB1 and RB20 groups, it has increased. So it gives a positive remark that by the use of RB1, we can treat neuroinflammation in a mice model. Next data is telling us about the beam walking test that is done for locomotor activity, balance, and the motor condition of the mice. In the first picture, we can see how firmly it is placed in the beam without any falling. Here we can see one hind limb is out of the beam. That means it's balancing. Thirdly, it falls from the beam and it is facing difficulty placing both hind feet on the beam, and lastly, falling from the beam. So these 4 conditions are marked while doing the beam walking test. The score of beam walking test is seen here in the LPS group is low. Similarly, the RB110 and 20 is increased. Time spent on walking is the highest. That is, LPS mice is taking more time to complete the pathway correctly. So in this way, we collect the data from the beam walking test. Next, we have the Western blot analysis report or data. So here we used to collect the data for the markers of the different phenotypes of microglial and astroglial cells, so that we can know that which glial cells need to be attenuated, which phenotypes of the glial cells are at its increased level or decreased level. Which kind of marker is being marked in the LPS groups, so that we can attenuate it accordingly. So here you can see that the LPS group has TNF alpha mRNA expression the most highest, which are attenuated automatically when there is administration of RB110 and RB120. Similarly, ionos also have attenuated. These two are the pro-inflammatory molecules which needs attenuation. That is, we need the reduction of this level so that the mice could have a good recovery. So it's giving a positive effect. Here we can see. And here we have the ionos or GAP-TH data, which also is seen that it is being attenuated properly by the use of RB110 and 20. Lastly, these are the more markers that we need to study to make sure that the pro-inflammatory molecules level is decreased or attenuated upon using RB110 and RB120. So C3 and CLF1, as I have discussed earlier, marks are the markers for A1 and A2 respectively. So A2 is a marker that repairs the cell. M2 and A2 are the repair type cells which are low in LPS. It's obvious. So it is increased when there is administration of RB110 and 20 to the mice. So it's telling us that by using ginsenosine, Panax ginseng, RB1 administration to mice, the neuroinflammatory model mice, we can attenuate or tune the glial cell polarization. So the study concludes that RB1 can attenuate chronic neuroinflammation induced by LPS mice, which may partially attributed to its ability to fine-tune the polarization of microglia and astrocytes. Further investigations include that polarization of glial cells must be done with diverse methods like pathological monitoring, staining of specific phenotypes. Secondly, detection of more markers. We have only a few markers that we have studied now, but further research is going upon to know more and more markers so that we can detect whose level is increased or decreased or needs attenuation so that we can cure the neuroinflammatory disease of the mice or the model organism. So detection of more markers like working with IBA1, which is a specific marker of microglia, mapped to expressed in axons and dendrites must be done. And also the sex differences related to drugs and models that if we must also know or be sure about to study upon the male model or the female models. So this is all about the study of the ginsenoside RB1, which attenuates lipopolysaccharide-induced chronic neuroinflammatory disease in mice by tuning glial cell polarization. Thanks for listening. And also, we will meet again with the next video presentation very soon. Bye-bye. Take care.